Hello, 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 hello. Notice I'm a little cold right now, hence like all my bundle up. I woke up and it was 62 degrees in the house. Oh man, look at this problem now, which is just too cold. Anyway, I gotta turn that off. Okay, so we are going to start on chapter 18 about gene regulation. And this video is going to be pretty short, talking specifically about prokaryotic gene regulation, or just prokaryotic genetic diversity. All right, and yeah, let me share my screen. Oh, Siri. Come on, iPad, do your thing. Alrighty. Okay. So we are talking about chapter 18. This is something that can be very confusing to students, this chapter. So reading the book in particular about operons would give you a nice little head start to this chapter, right? So you can go to chapter 18 and start reading about operons, which are involved specifically in prokaryotic gene regulation. And again, as always, the Khan Academy and Mr. Anderson have some great options. Again, the operons are very confusing. So these two about the lac and the trip operon are great resources. All right, so genetic variation is absolutely essential to the survival of a species. All right, and we have talked about, let's start here. We have talked already about how eukaryotes increase their genetic variation, all right? And that has to do through sexual reproduction. And in particular, we talked about three mechanisms. One, crossing over, which is in prophase 1 of meiosis 1. The second one is independent assortment, right? And that's how the chromosomes, how the homologous pairs line up at the metaphase plate of metaphase 1 of meiosis 1. And 3 is random fertilization, all right? But I want to point out that bacteria also have genetic variation, and we're going to talk about the mechanisms for that. So first off, a reminder, how do bacteria or prokaryotes reproduce? Right? So they just grow and divide, but it's not mitosis because it has no nucleus. Rather, it's called binary fission, just as a reminder. All right. So you have a prokaryotic cell, it grows, and then it'll eventually divide into its separate ones. And if you look, as you zoom in and look at the bacterial chromosome, we will see that that bacterial chromosome also is going to grow and replicate rather, and then get separated, right? So genetic variation allows these groups of organisms, and we're going to talk about this in the upcoming chapters. They allow groups to adapt to the changes in their environment, all right? So adapting has changes in the, basically in the genes that will allow them to have adaptations or ways that they can fit into their environment better, right? And that's by a process called natural selection. You guys have probably heard of natural selection. This was proposed by Darwin, and most students always come into the class being survival of the fittest. Like they know that term, survival of the fittest, but it's more than that. It's about not only surviving, but it's also about having babies, all right? So traits, the natural selection is that traits passed down from parent to offspring help that organism survive, right? Survival of the fittest, and reproduce, right? And that reproduction is really important because anything that has adapted to its environment better will have more offspring, right? And then that trait will become more common in a population over time. So it's about survival of the fittest and reproduction, or passing those traits on, right? So where does genetic diversity come from in prokaryotic cells? So again, we're going to talk about prokaryotes first, and we already talked about eukaryotes, crossing over, independent assortment. random fertilization. So now we're talking about how bacteria can change their DNA to increase genetic diversity. And it happens in 3 ways. 1, bacterial transformation. Remember the Griffith experiment with the S and R bacteria and how bacteria can uptake DNA from their environment. The second is bacterial transduction. And here, bacteria have bacteriophages or viruses that can carry DNA from one bacteria to another, right? And this reminds me of the Hershey and Chase experiment. Remember those bacteriophages infecting the bacteria. And then the third one is called bacterial conjugation. And this is where DNA is physically transferred between little tubes in the bacteria. It's kind of like bacterial sex, right? So the first one, again, bacterial transformation, the ability to uptake DNA from the environment, right? So here again is an example of the Griffith experiment. The R strain, you inject and you get happy mice, it's no problem. If you inject the S strain or the smooth strain, you get dead mice, right? If you heat kill the S strain, so if you take the S strain and you heat kill it, the bacteria or that don't infect the mouse. However, if you take that S strain, if you take that heat killed S strain plus the R strain, you get dead mice. So what that means is that the R bacteria took up the S DNA, right? So bacteria can take up DNA from their environment, incorporate it into their own DNA, thereby changing their genome, fundamentally changing their genome, right? So that's one way that bacteria can change their genetic makeup or their genome in order to become more, in order to increase their genetic diversity. The second one is bacterial transduction. Okay, remember viruses called bacteriophages carry DNA from one bacteria to another. So here example is our bacteriophage. This bacteriophage is introducing this DNA into this bacteria, right? So, as bacteria, as those viruses, then let me rephrase. So now this bacteria that has been infected, so this is an infected bacteria, it will have the instructions to make more bacteriophages, right? And that bacteriophages can incorporate both the bacterial DNA or the original DNA. So to make a long story short, bacteriophages are viruses that infect bacteria, and these viruses carry DNA from one bacteria to another, right? And remember, Hershey and Chase are the people that really discovered that viruses usually infect bacteria through chromosomal DNA, not the protein, all right? So bacterial DNA can be incorporated into genomes, and that thereby changing the genome and increasing genetic diversity. The third and final way that bacteria increase genetic diversity is through bacterial conjugation. I always think of this as like bacterial sex, where DNA can be transferred between bacteria through a tube called a pilus, right? See why I think of it as sex? It's got this long pilus. So bacteria or DNA is transferred between bacteria through the structure called the pilus. It makes a connecting tube between two bacteria. And now some DNA can be transferred from one bacteria to another, all right? And you'll notice in these bacteria that there's two kinds of pieces of DNA, which is interesting. We have the genome, which is the bacterial chromosome. The bacterial chromosome. And then we also have this little blue piece, which is called a plasmid. All right. So a plasmid is kind of a separate piece of DNA that occurs in bacteria. And we're going to talk more about plasmids as we go into our biotech unit. All right. So let's just, as a reminder, try to do this matching. All right. So match each type of gene transfer with its definition. And again, we're talking about ways that prokaryotics or bacteria increase genetic diversity. All right. So conjugation. So which one is conjugation? So if you remember, conjugation is the bacterial sex. So here, it's here. A bacteria transfers DNA as it's physically connected. A transformation is where bacteria can take up DNA from the environment, and transduction is a virus moving DNA from one bacteria to another. All right. And just as a reminder, eukaryotes have lots of ways, lots of genetic diversity from meiosis and sexual reproduction, right? And we've talked about the two ways here. We have one, crossing over during prophase 1, 2, independent assortment during metaphase 1 of meiosis 1. And the third way is random fertilization, all right? So what we're going to talk about in the next section is how do cells with identical DNA perform very diverse functions. So every cell in your body, every somatic cell, meaning the diploid cells, every one of those cells has identical DNA, which is crazy to think about, right? My eye cell has the exact same DNA as my toe cell, as my liver cell, my neuron cell. So how do those cells perform different functions, all right? And each of these cells has different functions based on what is going to be called differential gene expression. So what I mean by that is that different genes are activated in those different cells, meaning that those different cells make different proteins, right? So eye cells, for example, will make the proteins for cones and rods that make us able to see, whereas a toe cell will make proteins involved in, the skin formation and proteins involved in whatever big toe has to do, all right? And we'll talk about that. And that's really the crux of gene expression, is that every cell has the same DNA in a body, but they have different genes that have been activated or turned on or transcribed, making different proteins being made or different proteins being translated. All right, stop there. Stop there. Stop. In the name of love. Stop, calibrate, and listen. ICE is back with a brand new invention. Bye.